Prolific is enabling biopharmaceutical companies and CDMOs to extend their CHO production systems into the next generation of protein therapeutics. We have really thought about how to make all of this extremely modular. So whether it's taking our tools and engineering those into cell lines that we have in-house and regularly work with, or if it's taking those tools and engineering them into cell lines that our partners are working with, the cell line engineering that we are doing at Prolific is we're making changes that employ optogenetic control. What we are doing in the lines that we engineer is basically taking that genetic sequence that corresponds to the protein that we're interested in producing. We're taking that sequence and in a very precise and direct way, integrating it into the genome of a cell, putting that under the control of a promoter. That promoter happens to be driven by light, and we are also integrating in these transactivator proteins. What they do is when the right wavelength of light shines, they go and find that promoter and are driving that light-driven expression of our gene of interest. What this light-inducible control enables for us is it allows us to dictate when we turn gene expression on and to what extent. This can be really enabling in cell line development contexts because oftentimes what can end up happening is that there's a great deal of burden that's placed on cell line development. Maybe for a given protein that has multiple components, or if you're dealing with a complex antibody, what can be required is screening many clones of a given target to try to find the ones that have the right degree of expression or the right number of integrants that are producing the highest yield of that final product that you're really looking for. What our cell lines can enable is instead being able to use one single universal cell line that is able to give you that tunability to find where is that right level of expression that can give you that final product.